Alrighty, so this is Unit 4, Cellular Communication in the Cell Cycle. It's a lot of kind of different hodgepodgey stuff. It's 116 slides, so I'll go as quickly as I can, but it's kind of a lot of information. And so this is just the topics that we're going to talk about. There's a lot of these as we go through, but you can read those. This chapter 11, this was one that we, in the textbook that we have, didn't have everything that I wanted. So there's chapter 11 in our book and chapter 11 in the Campbell book, which is linked on my Schoology page, and then also chapter 45 in Campbell. And so if you want to go through and look at those, this is the chapter when we use both books. And so cells have to communicate in order to survive. So there are sort of short distances, long distances, prokaryotes, eukaryotes. So we'll talk about a bunch of different stuff. And so then we will also talk about the signal transduction pathway. The biggest thing that it does is it amplifies the signal, and then it takes a relatively small signal or little molecule and then turns it into what usually will be the cellular response. Usually will be we will transcribe the DNA and then make an enzyme or a protein or something like that. So this is just an example of the receptors are on the outside of these yeast cells, so they release factors, and then they can recognize those factors, and then they can, for this simple little example, then they can mate, and then they have sort of a combo baby. And then this is just some pictures. And then we have local and long distance, and we'll talk about those as we go through. This is just sort of an intro slide. But there are two ways that cells can get signals from one cell to another. Sometimes they're touching. We talked about the gap junctions and plasmodesmata in animals and plants, and so it's like a little tunnel, and so they just sort of scooch across because they are connected next to each other. And then what we'll talk mostly about in these slides is recognition and extracellular receptors. And then we have some things that travel short distances, some travel long distances. The one that we talk about the most with long distance is hormones. We have hormones that they get produced in one place in our body, and then it gets released out into our bloodstream, and then it affects other parts of our body, but it happens in animals as well, or I'm sorry, plants as well, and then all kinds of different animals, of course. And so this is just some pictures that go along with that. But there's two sort of short distance ones, or local distance ones that we talk about. It's called paracrine and synaptic. Synaptic should be easy to remember because it is like a synapse in the nervous system. And then neurotransmitters are something that would sort of bridge that gap. They're little molecules that go across. So when people take antidepressants, it's usually your body doesn't make those neurotransmitters, and so then the medication is the sort of substitute, and then the message can get sent from one little place to another little place. And then a little bit of longer distance is just showing how the endocrine cell may release the hormones into the bloodstream, and then it has a cell that it affects, and so that's called a target cell. And then this just intro stuff like before. And then there are three portions of a signal transduction pathway: reception, transduction, and response. So we get the signal, we translate the signal, and sort of we do some stuff with it intracellularly, and then there's something that comes from that. So I'll click the whole thing up there. And so the first little part of this is reception, the second transduction, third is response. So the signaling molecule we call a ligand. It is specific to the receptor. And so some cells have the receptor and some cells don't. So even though something might be out in your bloodstream, it won't affect the cells that it's not supposed to because the receptor isn't there. And then sort of a generic transduction pathway, but they go across, and then they affect, and then we have phosphorylation cascades, and then we have amplification of the signal, and then a cellular response. Like I said, usually a little molecule gets released, and then that goes into the nucleus, and then it transcribes. It's called a transcription factor, and then it goes in and transcribes and translates, and then we can go through protein synthesis. And then, so this I was talking about the little signal. It's called a ligand. It's a specific shape. It fits like a puzzle piece. There are three main ways that we can get an extracellular signal into the cell, and we have to do this on molecules that are water soluble. So think of the plasma membrane, the inside of the plasma membrane. These are lipids, and so it can't pass through. It has to have a way to get through. We'll talk a little bit later about lipid soluble signals or ligands. Those can go right through. They just go, and then they have an intracellular receptor. So these three things are on the outside of the cell membrane because the signaling molecule can't get through. So there has to be a way. So there's three that we talked about in class. G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. And so we'll go through them one at a time. And so the G protein coupled receptor, if you have your notes, maybe get we drew this on a really not great drawing, but hopefully sort of put it all into one place. But there is a G protein, and it's a thing that when the molecule comes up and then it recognizes it and it goes, and then it changes its shape, and then it activates other molecules in the membrane by changing in the shape, and then it triggers the transduction, which we'll get to in a bit. And so this just shows a picture of that process. And so we have the G protein coupled receptor, then we would have the ligand that would come in. We would take the GTP and GDP, and then it would go over, and then it would activate the little enzyme that is in the membrane. And then if we have an inactive membrane or inactive enzyme and it turns into an active enzyme, then that enzyme is going to do a job, and that triggers a intracellular response. And so then it all sort of releases and turns off until the next little ligand comes in. Receptor tyrosine kinases attach phosphates, and then the one cool thing, it says can trigger multiple signal transduction pathways at once. So this is the one that we tip the domino, and then the rest sort of make a whole big bunch, and then we get a huge response. And then a ligand gated channel, usually with it's also in the title, it's for things that are charged, and it has a little like trap door, and so it changes shape, and then the door opens, and then the thing releases, and then it goes back to the original shape. And so here's a little picture of that. And so the ligand comes in. If you can see how it says the gate's closed, and then you can see it's open, and then the stuff goes through, and then it releases, and then it goes closed again. And then intracellular receptors, it's usually lipid-based or small, or things that just can go across the membrane. So this just talks about a few of those. And then this just shows how testosterone or hormones in general are lipid-based, so they don't need a membraned receptor. So they have an intracellular receptor, so then it comes inside, the receptor's in there, does the whole, pretty much the same thing, transduction, and then response. And then this is the part that I was talking about with transduction. The point of transduction is to have a relatively small signal, and then we get a really efficiently produced big response. So that's pretty much what the transduction pathway does. And then I was talking about this, about like pushing the dominoes over. You can just read through that. And then a lot of times when we're talking about going from one of the reactions to the next little reaction to the next little reaction, we're talking about phosphorylation and dephosphorylation. And so we add or take off a phosphate group, and then it becomes a new molecule, which then becomes a reactant for the next thing, which then makes the next reaction go, which then makes another product, which then becomes the reactant for the next reaction, over and over and over and over and over again, just in general. It produces a product that becomes the reactant that activates the next thing. And so it's sort of like you have to wait for the thing to get produced in order for the next thing to start. And so this is just talking about that that process that there's a whole bunch of controls within the transduction pathway, within transduction segment specifically, as a kind of a checks and a balances or a on off or a make sure that the systems are working. There's a lot of places for stuff to go wrong, but there's also a lot of places that can compensate if something small goes wrong, rather than one thing and then the whole thing gets messed up. And so then this is just a oversimplified picture. It sort of shows you what I was talking about. And then one of the things that we talked about in class that's a little bit weird is second messengers. And so the ligand that comes in that sort of brings the message, that's called the first messenger. And then we have these little helpers that we need. We talked about cofactors and coenzymes, I think it was in Unit 2. And this is kind of the same concept, that we need these little extra molecules in order to make the other parts work. There's two big ones that usually that you'll need to remember: cyclic AMP and calcium ions. And so a little bit about those is the cyclic AMP, which is ATP with two phosphates removed, and then it's cyclic because it has the molecularly it's a circle. And then adenylyl cyclase is an enzyme in the plasma membrane that converts the ATP to CAMP. So if you flip back to the previous ones, it was when we talked about the G protein, the first one when we talked about the 3 extracellular receptors, and that was the enzyme that was in that picture. And then, so anyway, just to give that specifics. And then this is just a picture, which is the same thing that I was just talking about. And so we have that messenger comes in, and then that adenylyl cyclase, and then we change that, and we get ATP to CAMP, and then that CAMP is the second messenger that then enables that first step in the transduction segment to sort of kick off. And then it goes through the sort of all the different little reactions, and then we get a response. And then just another example is calcium ions, and it talks a little bit about that. And then not the last thing, but sort of the result of the transduction is a cellular response. Oftentimes, for what we talked about, it was a transcription factor. So we went through and we had this signal, and then we went through these sort of enzyme or these reactions, and then we had this little thing that came out. Well, that little thing goes into the nucleus, goes to the DNA and says, this is the part we're going to transcribe, and so then this is the enzyme that we're going to make. And so then just a little graphic of that. And then just a specific one, just to give you an example, that it can be a whole bunch of different phosphorylation, dephosphorylation reactions within that transduction. Just depends on what you're making. And so this is just an example. And then sometimes it changes the sort of the response or the result of it. We make transcription factors, but sometimes we can make things that give us funny shapes and stuff. So this is just an example. I just think the word is funny, so I left it in there. And sometimes they talk about schmooze, and I think it's a Dr. Seuss thing as well. And that's what it's talking about, is this weird little these weird little cells. And then two things are why we go through all of those steps within transduction. The biggest one is amplification of the signal. A very small signal gives us a big response. And then the specificity of the signal, because the path might be going this way, and then we branch up this way, and then we branch off, and then we branch off, and we branch off, and we branch off. And so we can get to a very specific molecule. Rather than having a pathway for this, and a pathway for this, and a pathway for this, we can have a sort of common pathway, and then they branch off and specialize. If that sort of makes sense. Kind of sounds like a cladogram, but we didn't talk about that. But it's sort of like, if you think about like evolution of an organism, sort of the we're sort of on the same track for a little while, and then we branch off. And then this just talks a little bit more about amplification. So it's pretty simple. And then, this I talked about. But it helps us to get a very specific, efficiently made, sometimes transcription factor, oftentimes, and then, or whatever else the pathway might be producing. And then sometimes our body puts things in the same spot, physically in the same spot, because it's more efficient, and they're on what's called scaffolding proteins, and so they sort of hold stuff in the same location because they're all going to get used in the same process, and so it makes the whole process more efficient. And then there's just a picture of it. And then sometimes we need to turn it off. What often happens is when we make enough, it gets turned off because the molecule pops off. So we'll go through these and then the next little section. And then sometimes we have when the pathways don't work. And so then, this is just a little bit about cancer. And so we have, whenever the signal isn't listened to, maybe in the mitosis, did we make the RNA or the DNA correctly? Did we make all our cell organelles correctly? Our body's supposed to listen to those signals, but sometimes they don't, and then we can get abnormally growing cells. And then this is just some vocabulary, so some oncogenes, proto-oncogenes, growth factors that sometimes it doesn't listen to the stop, and so it just keeps on going. And then we talked a bit about things. Tumor suppressors is pretty easy to understand. BRCA1 and BRCA2 are the ones that are breast cancer genes. We talked a bit about those, but pretty easy to understand. And then benign and malignant are, benign is you don't want them, but they're usually okay. And then malignant, like mal in Spanish and French, means bad. That's what comes from the same root word. So the malignant cancers are the ones that are bad. And then this is just talking about some extra stuff about cancers and sort of details about that, and then another vocab word at the bottom. So metastasis is when it sort of starts in one location and then breaks off into another one. I think we had a question on one of the tests that talked about the cells adhering to one another, or the lack of adherence. So like an adhesive. And so if they don't stick to where they're supposed to and they sort of float off to a different spot, that is one of the ways that metastasis occurs. And then that's just a little graphic. And then some details and some how we can not do it. Don't smoke, don't go in the sun without sunscreen. Make sure you get pap tests, maybe breast self-exams, colonoscopies to help detect it early or prevent it in the first place. And then just some pictures. And then feedback for the systems is how we sort of turn things on and turn things off. So this is chapter 45. And so hormones are the long distance signals. We make them long distance in our bodies because cells next to each other or cells not next to each other, then it's considered long distance. And so then there are 5 long distance molecules that I want you to sort of understand or be familiar with. And then endocrine glands are oftentimes hormones. And then they release whatever they're producing into the body, and then exocrine glands are sort of out of the body or out of the wherever they're located. And then this talks about some words that we already did talk about, so I'll go through these quick. And then again, same pictures. And then pheromones are one of the only chemical signals that go outside of our body, and we talked about it's sort of a smell or like a something that you detect in a weird way. So it would be like from person to person or animal to animal. And then we talked a bit about these, that lipid soluble pass through easy, water soluble don't, so then they need a receptor. And the same thing here. And then the same hormone may have different effects on target cells, and so it might affect skin cells differently than sex organ cells. And so we might have like hair growth versus egg production versus milk production or different things like that. And then there's negative feedback, which is when we talked about this, it was when something sort of turns something on and then it has like a partner that turns it off. And so the analogy I used in class was like a thermostat in your house. Like you want it about 70 degrees in your house. So if it gets too hot or if it gets too cold, it sort of turns on the AC or turns on the heat or turns it off when it gets, so it's sort of like that. And so this is just talking about a specific example with insulin. So insulin gets rid of sugar or breaks down sugar, and glucagon increases sugar. So it just talks about how those work, and then we'll talk a bit about diabetes. And then there's a couple kinds of diabetes. Just be familiar with those. In that diabetes, type 1 is usually you have it as a child. Usually you are born with it. And then type 2, some people develop over a lifetime. And then another example is oxytocin, which is the hormone that right after baby's born, it's the one that helps during childbirth, that there's uterine contractions, and then when the baby's breastfeeding, it helps, or it's when the milk is released, and then the more a baby breastfeeds, the more oxytocin is released, the more milk is produced, the more the baby eats, the more oxytocin, the more milk is produced. So negative is we're keeping sort of we want a pretty narrow set of conditions like temperature, body temperature, and then positive feedback is we make it and then we make some more, and then we make some more, and then we make some more, and so it gives a bigger response. And then this is just talking about in our body we usually use negative feedback loops, but there are some very specific ones, and like childbirth and milk production and different stuff like that. Then we have a positive one. And so then all of these things, all of our enzymes, all of our chemical reactions in our body, it's all done to regulate a certain environment, a certain pH, a certain temperature, a certain enzyme level, all of it. So we have to regulate our bodies so that they function properly. And so then we have just a couple of examples of hormones, specifically with males and females and humans. And so then we get organs, we get what are called secondary sex characteristics, and so we like facial hair or increased muscle mass or different stuff like that. And then just some other examples with women. And now we're going to switch gears into mitosis. I'm going to go a little faster through this because I think it's pretty easy. And so we talked a bit about Henrietta Lacks. If you forget, go back and watch the stuff. She's someone you should know. And then a picture of her. And then we have the cell cycle is a big cycle. It's a big, I drew it as a circle in class. And so the first sort of 2/3 of it are interphase. And then we have mitosis, or the M phase, and then cytokinesis, or that very last little part when they split into two. And so there's 3 sections of interphase, G1, S, and G2. The most significant is the S phase, when DNA replicates. And then there's a lot of checkpoints, several checkpoints, maybe not a lot, that occur during the cell cycle. Did we make enough cell membrane? Did we make enough cytoplasm? Did we replicate the DNA properly? Are we ready to sort of go through the cells, they're the mitosis, the mitotic divisions? If those aren't listened to, sometimes they can develop abnormal cells, we might not have made it correctly, but our body didn't listen to that signal, and so then we make it. We might make too many. We might not shut it off when we're supposed to. Different stuff like that. And then mitosis is considered asexual reproduction. We make an exact duplicate. There is no girl skin cell and boy skin cell. We'll go through the steps. And so then just a picture. And then we have homologous chromosomes. So homo means same. And so you have one from mom and one from dad, 23 pairs. And then this just talks about the 4 phases. And so then we have 2, they replicate, they split apart, and then they split into 2. And then we have, I talked about checkpoints already. And then this is just showing you a little graphic about the G1, S, and G2. These are indicative of the time spent. Think of that circle like a day. And so like the vast majority of the time is spent in interphase. A huge part of that is G1. Another huge part is S. A little smaller, but still significant, is G2. And then mitosis happens relatively quickly. And then this just talks about the 4 phases. We go from chromatin to chromosomes because only during the mitotic divisions, because we're not going to take the energy to condense something if we don't need it to. So for the most part, remember when we looked under the microscopes, for the most part the cells were in interphase. And then this just shows some pictures, and then we looked at both of these in class, so hopefully you remember those. But prophase is the beginning. The chromosomes condense, we sort of start to get organized. We get rid of the nuclear envelope. We have to get rid of that because if it's going to spread out and sort of do some divisions and spread, that membrane would constrict it, and so that doesn't happen. It comes back at the end. A centrosome is where the little centrioles are. It's a little portion of the cell. The spindle is just the stringy parts that come out from the centrioles that then attach to the centromere and then pull. Metaphase is when they line up, anaphase is when they go apart, telophase is when it pinches down the middle. And then this is just a picture of those things. Hopefully these look a little bit familiar because we did look at them in class. And then cytokinesis is the last little step when they pinch in 2. And then there's 2 words that I want you to remember. A cleavage furrow, and animal cell is flexible because of the cell membrane, so it cleaves in the middle and then it separates into 2. That's called a cleavage furrow. A cell, a plant, is rigid because of its cell wall, so it builds up like a little membrane or little bubbles in the middle, and then it's called a cell plate, and then that's where the cell wall forms. And so then this is just a picture of animal cells in a cleavage furrow. Here's one with plants, and it's making a little that little cell wall in the middle. And then this is just sort of an extra thing because it's talking about cell reproduction. Is when we go through mitosis and we copy a previous cell, we're copying chromosomes. And so there's a little thing on the end of our chromosomes called a telomere, and it's sort of like a little cap. I think of it like an aglet on a shoelace. It's a protective thing, and it makes it so that when we're doing the copies, it doesn't sort of wear and tear too much. And so, because if we wear and tear our chromosomes, we're losing genetic material, and then that's not good. And so that if we were to put a fluorescent marker on them, then they would glow like that, which is pretty cool. And then it's just talking about the structural part that they don't copy, they don't do anything. It's just a protective thing. They get short, and then we trigger cell death. And then this just talks a little bit about stem cells and why they're cool and useful and interesting. And then this it was a lab that we did, and we could do this. So if we take 20, 30, 33, 34, 35, 36, and then we decided 20 divided by 36, 10 divided by 36, this would give you an approximation of how long they're in there. So there's a lot of time here. This is maybe a little bit, should maybe be a little bit higher, but spends a good amount of time in this, and then these happen relatively quickly. So we could mathematically look at cells, which is what we did in class, and then do a little table and then calculate, and then this is sort of how the pie chart is is produced. And so apoptosis is normal, programmed, healthy cell death, because there's only, it's like when you make a copy of a copy, eventually you can't keep on making copies of the copy. And so you got to, it's just our cells only go through so many cell divisions before they naturally just dissolve and get discarded by our bodies. We just recycle them. But that sort of triggering of that is called apoptosis. And so it makes these little like bubbly parts on the outside. I'll show you a figure, but that's sort of a healthy cell on the left and one that's going through apoptosis on the right. And then this is just talking about the sciencey stuff behind it. But if we have a receptor and it sort of hangs out there until it is it's time. And then death signaling molecules, like the Grim Reaper of the cell, comes in, activates that, and then it starts the process, signal transduction, and then it goes and it makes these funny little bubbly parts, and then it looks like that cell that I showed you in the previous, one of the previous slides. And then just a couple other things is sometimes apoptosis is normal, sort of like a scheduled thing, and sometimes it happens when the body detects that something went wrong. So we're at the checkpoints, or we're at we didn't make the DNA correctly, or the DNA somehow got damaged, or we have the proteins were messed up, and so then we're just like, okay, we're going to just scrap this, we're going to start over. And so that would be a time when we could apoptosis would be really useful. And then last one is whenever we have to sort of get rid of pieces and parts that aren't supposed to be there, sometimes it's a good thing. Sometimes it sort of goes in overdrive, and they think there is a link with Parkinson's and Alzheimer's, that maybe the apoptosis is going a little bit too well. They're not too well, but it's working too much, and then we get disorders that come along with that. So that's Unit 4.